Ladies and gentlemen, our first guest has arrived today. Kevin Walton is in the green room, so we're going to be bringing him on just momentarily here. So again, Kevin is working on some pretty cool stuff. Let's just take a quick look at Baseimmune here. Preclinical, working on some really cool applications for vaccines inside the fibrosis space. So let's bring him on. With no further ado, let's bring him into the studio. Kevin, welcome to the show.
Hey, how are you?
We're doing well. Good to see you.
Thanks for having me.
Glad to. All right, so I guess just like right off the base, can you give us a little bit of background on Baseimmune? What's kind of the big problem you're going after here?
Yeah, Baseimmune, we're tackling fibrosis with our active immunotherapy approach. That's based on a computational platform where we can multiplex epitopes on an antigen. And we're going after idiopathic pulmonary fibrosis, or IPF, first. It's a devastating disease where scar tissue progressively replaces healthy lung tissue. It's hundreds of thousands of patients globally. The life expectancy upon diagnosis is 3 to 5 years, so it's a devastating disease. There's been decades of research, and there's only been 3 drugs approved that only slow the slope of decline. Nothing halts or reverses it. And then there's been a lot of failures in the clinic. And I think that's kind of where we think we can be quite differentiated. We think a lot of the failures are due to everything going after a single target. But the issue with the biology of fibrosis is that it's inherently a network-driven, redundant disease. And so our computational platform allows us to go after multiple pathways with the same active immunotherapy.
Super cool. So when I think of it's an anti-fibrosis vaccine.
Yes.
When I think of vaccines, I think of my immune system killing something. What is the immune system killing in this case?
Yeah, so I'd say it's a therapeutic vaccine, so it's a little bit different. It essentially uses your immune system as the generator of antibodies, polyclonal antibodies for multiple targets. From the same vaccine to go and then block and prevent the progression of fibrosis. And so the goal is...
Okay, that makes way more sense.
The ultimate goal, right, would be to halt the progression. But kind of like if you look at it as... Fibrosis is very much analogous to cancer. And so if you look at it in that respect, if you're able to significantly slow the decline, you're then going to add many, many years of life. Right now, the 3 drugs out there, it's not a huge meaningful improvement, and the tolerability issues are such that many patients drop after less than a year on treatment.
Okay, so are the targets pathologically associated targets, or are they things that we would have tolerance to?
One of the things we have to do with our synthetic epitopes is to break tolerance. So we need to make them different enough to break tolerance, but similar enough that you still...
Cross-reactivity.
Exactly.
Yeah. Okay. So okay. Now it makes way more sense. There are a lot of companies who are doing this is much more elegant than what other companies do. Like I know companies like Vironexus, for example, they just use an AAV derived or AAV mediated like gene therapy to have hepatocytes produce a bispecific antibody for cancer therapy, for example, where the therapy is meant to have your body producing some kind of therapeutic antibody, right? This is kind of the same thing essentially, but this is much more elegant and immunologically like nuanced.
Yeah, it's similar to, you have like the Moderna and Biotechs of the world doing the cancer therapeutics, cancer vaccines. You actually have quite a few vaccines going after Alzheimer's. You also have some for things like other autoimmune disorders like lupus. You have some for cardiovascular as well. And so I think the modality, or maybe not modality, but the approach is not new. But I think what we're able to do is we're able to do this in a way where you can go after multiple pathways. And especially for something like fibrosis, where it is pathway driven and shutting down one pathway, other pathways compensate for it. I think that's where we can see hopefully a much more transformative impact on patients.
So that's super elegant. I love it. Is there a reasonable risk of toxicities? Since you're breaking that tolerance, could you get some kind of like extended cross reactivity that damages tissue?
Yeah, I think the biggest safety question, right, is right now in development, a lot of the candidates in like phase 2 are monoclonal antibodies. And so you infuse the patient with monoclonal antibodies, there's a half-life, and then they go away. Now the bad part is, right, if you want to keep having a impact on the patient, you have to do chronic infusions. Whereas for our approach, you would likely have to get boosters, but it'd be much less frequent, and it's a booster, not going into the hospital, getting hooked up to an IV and having that infusion. The thing though is how do you turn it off?
Yeah.
There's been ways shown that you can. I think also the way that your body is just innately operates is that it doesn't keep producing it when it's gone. If you think about other therapeutic vaccines, like in Alzheimer's, I think you have some phase 2 assets, and it shows that there isn't necessarily a safety issue. You also show that if you look at some of the monoclonal antibodies safety issues, getting those infusions, you don't have a lot of necessarily bad side effects elsewhere in the body. But that is something that we will have to obviously monitor closely.
Sure. Yep.
Sure. Yep.
So with previous approaches that have gone after this disease and they haven't been really all that successful, is it partially due to the fact that they're going after just one pathway, and so you kind of just get rerouting around it?
Exactly. Everything right now is really single target, and so the belief, right, is, and if you maybe take a step back from an evolutionary standpoint, it makes sense. If your lung gets damaged, you want to make sure your body can find a way to heal it. Now, fibrosis, pulmonary fibrosis, is really just the dysregulation of that mechanism where it overheals and it keeps producing that scar tissue, which takes up the healthy tissue. And so taking one pathway, one target, the other pathways will compensate because it's been dysregulated. So your body keeps thinking we need to produce more to heal. Okay.
Interesting. And then I guess like just in terms of like the early data that you guys have seen, like what kind of got you excited here? Like what was the thesis where you're like, oh shoot, we've got to make this into a company. This is something that patients need.
Yeah, so it's actually interesting. So Baseimmune's been around a little bit longer, and so we started off looking at infectious diseases based on our platform, and so we were able to generate some compelling data in coronavirus, where you can actually make one vaccine that can work across multiple variants. So instead of saying what's the next variant, you can actually have something that lasts longer because of the ability to go after multiple targets. We also had some compelling data separately in malaria. And then we wanted to pivot into chronic diseases. And when we were looking at the wide landscape of chronic diseases, there was, I'd say, 5 or 6 that really boiled to the top. And we chose fibrosis over the other ones specifically because I think it is really custom fit for the disease and the disease biology. And I think you have a number of programs that have worked a little bit in phase 2. And so you can see, okay, if you go after that target, there is an efficacy signal that does help impact fibrosis. However, right, it likely isn't doing enough on its own. And so by looking at kind of the validated targets, and when I say validated, I mean that you have some type of efficacy signal. We think that if you take enough of those and put them together in our approach, you can have a much more transformative impact. The issue of infusing kind of cocktail monoclonal antibodies is you're going to have a lot more tox issues, and you're going to have different issues there that we don't necessarily encounter with ours. And so far, what we've been able to show is in silico, we still express the antigens that we're trying to. They bind to clinical mAbs, so we can make sure the epitope's intact. When immunized, they're breaking tolerance, so they're generating antibodies, and that's an important factor based on the earlier comment from Jeff. And then we also have shown that we can reduce collagen in some bleomycin models. And so, working currently through some in vivo models as well, but so far everything's been very encouraging in the last 9 months.
Nice.
Sounds good. So I'm very curious, and tell me to be quiet if I'm approaching any kind of proprietary information. But I'm curious. You mentioned that the biology of fibrosis was such that it was amenable to this approach. Does it have to do with the actual the ability to use this multiplexing discovery engine to find candidates that will work immunologically and break tolerance? Is it is it kind of like in that realm, or it's something different?
I'd say it's a little bit different. So we do have some ways to find novel targets, and that can kind of work across diseases. Right now for fibrosis, we're specifically focusing on more known targets that have shown some type of efficacy signal in phase 2. And that's because we're taking a lot of technological risk. And so how do you balance that with the biological risk?
Sure. Yeah.
And I think the technology is especially custom fit for fibrosis just because of needing to go after multiple pathways that would be really hard to do in other approaches and combination therapies. And I think it's the ability to kind of, the one thing we've done a lot of work on is looking at the different pathway drivers of fibrosis, what targets are kind of incorporate or under each one, and how can we make sure that we ultimately get a therapy that's going to hit at least 2 or 3 of those.
Yeah. So I don't want to divert the conversation too much, but this is, it's approaching one of the things that I have a very passionate opinion about, which is cancer vaccines. I'm going to assume that the biology is amenable also in fibrosis more so than it is in cancer, because the fibrotic like tissue is not so aggressively immunosuppressive and difficult to have an immune response there. Is there something there, or am I mistaken?
Yeah, I think the tumor microenvironment is going to obviously be very different than like the extracellular matrix environment from fibrosis. I think the other thing is that you also on tumors, and I'm not a biologist like you are, so I will defer to you on that. But I think within oncology, like I know the cancer vaccines, they're really looking at a lot of the neoantigens that are specific to the tumor, whereas fibrosis is really an inflammation. And if you think about it, it's an inflammatory type disease where it's your own cells, right? It's not neoantigens that are unique. And so I think that is a big difference, is how do you go after kind of self targets, proteins.
Got it. Yeah. One of the things, I mean, I have a lot of opinions about the cancer vaccine space. One of them is that we basically treat the cancer vaccines like prophylactic vaccines, and it's just not the same type of physiology. Like the biology just is not the same between like protecting against chickenpox and killing an active tumor. And the technology doesn't seem to really appreciate that as much as it should. But it seems like fibrosis would be a different beast altogether, and the problems with cancer vaccines wouldn't really apply to a fibrosis vaccine.
Yeah, no, I think that's great. I look at more of how would you go after, say, a neurodegenerative disease, cardiovascular disease, pain, things like that, where it's more kind of looking at like self proteins versus the tumor microenvironment.
So,
Nice.
So I want to pull on one thread that you just mentioned earlier, right? You're going through looking at these different chronic diseases. 5 or 6 surface to the top. Obviously, fibrosis is the top candidate, the largest unmet need. But I would be curious, like what other disease indications surfaced in that initial discovery phase?
Yeah, I think one that I'll share 2 that I got pretty excited about as well. One of them is pain. And I think it's a very similar thing. There's a lot of drivers of pain.
Pain vaccine. Oh my gosh. Okay.
What in the world?
But, I think the thing I would challenge there is think of it more of an active immunotherapy. So not necessarily you're taking it as a preventative prophylactic, but it's more therapeutic, where if you were to get chronic migraines, you would take this, and then that would help in terms of the intensity, the severity, but also the frequency. And
but imagine the marketing opportunity I mean, even if the biology is technically not like this, imagine the marketing opportunity of providing a vaccine against pain. That would just kill in public.
It would be, you would have to isolate what are the main drivers for like migraines in terms of just like the biology,
of course,
and then
develop
something that goes around that. But I think, I have a headache, I can go and take, acetaminophen or ibuprofen, and that helps with the headache. But that's not necessarily like the blockers of the causes of it. And it's also not a migraine. If I have a migraine, that's a different cause. And so if you can isolate certain routes and then go after those, that's huge. And it's typically going to be multiple routes, right? And same for cardiovascular diseases. So you have a lot of patients who are taking multiple drugs to help essentially your cardiovascular health. How can you actually design one that's going to be much more powerful and doesn't have, you're not taking assortment or a cocktail of
Right.
small molecule drugs and pills just to try to like maintain a healthy heart. And especially with, I think, the last few years, there's been a lot of research on new targets, and you hear a lot more like, LP little a, things like that.
Yeah, I think the combination of that aspect where we're just like learning more about biology as time goes on, right? Combined with AI and the ability to like generate these new, like you guys, I mean, like, a multiplex vaccine that breaks tolerance, like that would be basically impossible 15 years ago, right? But now with these really powerful computational models and things like that, I'm not super sure how yours works, but it becomes much more feasible to do these things.
Yeah. I unfortunately I can't tell you how it works, but it's I
won't ask.
Yeah, well, but you're right. I mean, we're able to down select from hundreds of thousands very quickly using our machine learning program.
Yeah.
Excellent. So we actually have a comment here just from a bio briefcase here. You made antigen-specific T cells. Great. Can they infiltrate a tumor? Can they overcome immunosuppression? Can they avoid anergy? It's just kind of in the context here of the problem of cancer vaccines. They're also saying that they love the content. Well, thank you at bio briefcase. We appreciate you coming. So I guess maybe
we know him.
Do we? Perfect. I'm blanking. My brain is mush. My child was up at 4 o'clock this morning. But yeah, that's an interesting.
So we have a T cell. So our platform, we have a B cell approach that's like a VLP protein, and a T cell approach that's mRNA LNP. For the mRNA LNP, I haven't talked much about that approach, but that's more of a pain fibrosis approach where was listening to a show earlier. It's a little similar to resolution therapeutics, but it's different because it's not cell therapy. It's still active immunotherapy or a therapeutic vaccination. And what it's doing is it's essentially allowing T cells to go and destroy the fibrotic tissue. So it's a little bit analogous to, I think, the macrophage approach, but also different. We've not done anything in oncology, but I think that there is the potential for the technology being applied there. But like Jeff was saying earlier, there is also differences in terms of just disease biology, and I think that we are not oncology experts at the company, and so we're more immunology experts, and so that's something that we've stayed away from on purpose.
Yeah, oncology is a different beast, especially when it comes to vaccines. It's a tough. I would wait before you guys even have any kind of inclination to think about doing oncology vaccines. Just wait until we get some positive results from Moderna or whatever is going on over there.
Probably later
otherwise,
this year they'll have their first big pivotal readout. It'll be interesting. I think there is positivity, but how much of that is? Yeah, it's tough to dissect out because it's also a combination with Keytruda. So true.
Yeah, Keytruda is 80% of the response. So,
but an amplifier and a meaningful improvement in life expectancy is also a great thing. But
I think you're going to have some kind of like adjuvant on top of the adjuvant when it comes to those things. Keytruda being one of those things, including like cytokines and all kinds of stuff. So it's a whole like.
It's also very different too, because there you have to analyze the tumor for the neoantigens. You can't just say here's the 3 or 4 big targets or pathways and do it for everyone. It's a unique individualized therapy for every patient, which is a very different ballgame.
Yeah, you would need to have like a similar system to yours, but have it be like basically real time.
Exactly. Yeah. But we have shown in malaria a T cell approach that's been fantastic. It's performed much better than most of the academic models because of the ability to go after the targets and be much more specific. So the T cell approach is something that is promising, but that's kind of more of our second program after the IPF.
Yeah, fantastic. I like the antibody approach a lot. That's like super. There's so many companies doing that with like autologous B cells and things like that. So to have it be like a vaccine approach, that's really interesting. So
yes,
we're excited.
Yeah,
nice. All right, so Kevin, I guess just the last question here would be, what's next for Baseimmune? What should people be watching out for?
Yeah, we're generating some in vivo efficacy data right now in mice, and then we'll look to use that to go after a multi-target approach to essentially design and optimize a lead drug candidate. Hope to get to proof of biology and IND enabling in the next 18 months. And then
nice.
go from there.
Excellent. All right. So there is a customary question we try to ask each guest just at the end here, which would be what's being slept on in biotech that's going to be really big in a couple of years? Ideally a pithy response, 30 seconds or less.
Sure. I'm biased because we're producing active immunotherapies, but I think
Everyone
really
is.
the immune system as a drug manufacturing platform that you can utilize. I think the biology that we have inside of ourselves is amazing with the immune system. It's an incredibly sophisticated drug factory. And so it's really, how do you exploit that for going after these diseases?
So you mentioned that you're a bunch of immunology nerds. That was great. So you mentioned that you're not oncology guys, but you're immunology guys. I'm also an immunology nerd, and I totally agree with that sentiment. Utilizing nature has gifted us with this incredibly complex system that is designed to heal our body. We should be using it to heal our body. And we're only now being able to uncover, or I guess exploit those things with the advances in technology. But I think that we're only scratching the surface, and I love that answer. It's great.
Excellent.
Thanks.
Well, Kevin, we really appreciate your time. We know we went a little bit over here. Thank you so much for coming, and we look forward to seeing your progress into the future here. Thanks so much.
Thanks so much for having me. Thank you, guys.
You are most welcome. All right. Thanks so much for coming, Kevin.
If you like this video, subscribe for more. We've got a boatload of content coming. Also, who should we talk to next? Drop your suggestions down below.